Picture this. You are a conservation biologist. Your entire career has been devoted to preventing animal species from disappearing forever, and you have seen enough to know exactly what the numbers look like when a species is in serious trouble. You know that a population of 10,000 breeding individuals is considered dangerously small for a large mammal. You know that below a few thousand, inbreeding depression begins compounding other threats. You know that populations in the low hundreds enter what biologists call the extinction vortex, a self-reinforcing spiral where small population size creates vulnerability, vulnerability causes further population decline, and the decline makes the next crisis even harder to survive. You are sitting at your desk one afternoon, reviewing a new population genetics paper, and you encounter a number that stops you completely. At certain points in the deep human past, the estimated effective population size of the entire human lineage, the number of breeding individuals whose descendants would eventually survive to produce all living humans, may have dropped to somewhere between 1,000 and 10,000 individuals, possibly lower. Some estimates for specific bottleneck events place the ancestral human population at numbers that, if applied to any other large mammal today, would have that species on every endangered list in existence, surrounded by conservation emergency protocols, and the subject of desperate international rescue efforts. The species in question, in those moments, was us. The fact that you exist, that any human exists, is the product of a chain of survival that came close to breaking at least once and possibly several times across the deep past. The story of how close humanity came to never continuing is one of the most overlooked and most humbling narratives in all of evolutionary science. And the evidence for it is written in extraordinary detail into the DNA of every living person on Earth. Population genetics is the discipline that uncovered the evidence for human near-extinction events, and understanding how it works is essential for understanding why the evidence is considered credible rather than speculative. Every human alive today carries approximately 3 billion base pairs of DNA, inherited from two parents, who inherited theirs from four grandparents, and so on back through the generations. As you trace the lineage backward, the number of distinct ancestors grows, theoretically doubling with each generation, until it exceeds the actual number of humans who were alive at any given time in the past. At that point, ancestry lines must converge. The same individual appears multiple times in different branches of your family tree. This mathematical reality means that everyone alive today shares common ancestors from surprisingly recent periods, and that further back in time, the effective ancestral population was far smaller than the actual population of humans living at that time. The genetic signal of a population bottleneck, a period when the breeding population was severely reduced, is detectable because it produces specific patterns in the distribution of genetic variation in descendants. When a large population is reduced to a small number of survivors, much of the genetic variation in the original population is lost. The survivors carry only a subset of the alleles present before the bottleneck. Their descendants, even after the population expands again, show reduced genetic diversity relative to what would exist without the bottleneck, and the pattern of that reduced diversity carries specific statistical signatures that population geneticists can detect, date, and roughly quantify. Modern humans have substantially less genetic diversity than would be expected for a species of their current size, age, and geographic distribution. Chimpanzees, our closest relatives, present in much smaller numbers today, and restricted to a fraction of Africa, have greater genetic diversity than all 8 billion living humans combined. This extraordinary reduction in human genetic variation is the smoking gun that points to one or more severe population bottlenecks in the human past. The Toba supervolcano eruption is the most frequently cited candidate for the most severe human population bottleneck, and it is worth examining in detail because the evidence around it is both compelling and contested in ways that illuminate the difficulty of reconstructing events from 74,000 years ago. Approximately 74,000 years before the present, the Toba volcano on what is now the island of Sumatra in Indonesia erupted with a force that dwarfs anything in recorded human history. The eruption was the largest in at least the past 2 million years, and possibly much longer. It ejected approximately 2,800 cubic kilometers of material into the atmosphere. For comparison, the Mount Tambora eruption in 1815, which caused the year without a summer and widespread crop failures across the northern hemisphere, ejected approximately 160 cubic kilometers. Toba was roughly 17 times larger. The immediate consequences were devastating across Southeast Asia, where thick ash deposits, in some locations meters deep, covered the landscape and almost certainly destroyed vegetation and animal life across enormous areas. The longer-term consequences through what is called volcanic winter involved the injection of sulfur dioxide into the stratosphere that reflected sunlight globally, producing years of reduced temperatures and agricultural devastation. Though no agriculture existed at the time, the impact on plant and animal food sources would have been equivalent in effect. The genetic bottleneck hypothesis proposes that this catastrophic event reduced the global human population to its minimum, possibly to somewhere between 3,000 and 10,000 individuals distributed in isolated refugia where conditions remained survivable. The support comes from the convergence of the genetic bottleneck timing estimates with the Toba eruption date, from the near absence of human archaeological evidence in the period immediately following the eruption in some regions, and from the logical severity of the environmental impact. The controversy around the Toba catastrophe hypothesis is substantial and scientifically legitimate. Archaeological evidence from South Africa and India suggests that human populations survived in those regions without dramatic discontinuity across the Toba eruption horizon. Some sites show continued human occupation before and after the ash layer with relatively minimal disruption. Critics argue that the hypothesis overstates the global demographic impact, that humans in coastal and equatorial refugia may have survived the volcanic winter with less severe consequences than the hypothesis assumes, and that the genetic bottleneck may predate Toba or reflect multiple distinct events rather than a single catastrophic reduction. The most balanced current assessment is that Toba likely caused significant regional population reductions and may have contributed to a genuine global bottleneck. But whether it was the sole cause of the genetic bottleneck, the primary cause, or simply one of several contributing events in a more complex demographic history remains genuinely unresolved. The genetic evidence for population bottlenecks in the human lineage extends well beyond the Toba era event and into the deeper hominin past, where multiple episodes of population stress and demographic collapse appear in the genomic record. A study published in 2023 using a statistical method called fast infinitesimal time coalescent process analysis found evidence for an extreme population bottleneck in human ancestors occurring approximately 800,000 to 900,000 years ago, dramatically predating Toba and falling in the period when the evolutionary transition from earlier Homo species to what would eventually become the ancestors of modern humans, Neanderthals, and Denisovans was occurring. This inferred bottleneck appears to have reduced the ancestral human population to approximately 1,280 breeding individuals at its minimum, maintained over a period estimated at around 100,000 years. If accurate, this represents a near-extinction event far more severe than the Toba bottleneck hypotheses suggest for the later period. A population of approximately 1,200 individuals sustained over 100,000 years is a population balanced on a biological knife edge, small enough that random demographic fluctuations, disease events, or localized environmental catastrophes could have eliminated it entirely, with no remaining population elsewhere to recolonize lost territory. The timing of this deep bottleneck corresponds to a period of significant climatic deterioration in Africa. The transition into what is called the Middle Pleistocene, characterized by longer and more severe glacial cycles, expanded African aridity, and significant reorganization of African ecosystems. Forest refugia would have contracted, large prey animals distributions would have shifted, And the specific mosaic environments that seem to have supported early hominin evolution would have become more restricted and less predictable. The combination of environmental deterioration with the small population sizes implied by the bottleneck creates a scenario where the emergence of later, larger brained Homo species was, from the perspective of the deep past, genuinely unlikely. The concept of genetic diversity loss during bottlenecks has practical consequences for living humans that are worth understanding, because the story of near extinction isn't just historically interesting. It is biologically present in every person alive. Low genetic diversity has well-documented consequences for populations. Inbreeding depression, the expression of recessive deleterious mutations that accumulate in small populations because they are no longer diluted by the genetic variation present in large populations, can reduce fertility, immune function, and developmental stability across generations. Species recovering from severe bottlenecks carry elevated frequencies of the genetic variants that happened to be present in the small surviving population, including deleterious variants that would have been held at low frequency by selection in larger populations. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Modern humans show several patterns consistent with a history of severe bottlenecks. Certain genetic variants associated with disease susceptibility are present at higher frequencies in all human populations than would be expected if the ancestral population had always been large. Immune gene diversity in humans, while not trivially low, is substantially lower than in other primate species with much smaller current populations. The specific mitochondrial DNA lineages carried by all living humans trace to a single ancestral female, the so-called mitochondrial Eve, who lived approximately 150,000 to 200,000 years ago. Not because she was the only woman alive at that time, but because her was the only maternal lineage that survived to the present without interruption. All other mitochondrial lineages alive at that time have since gone extinct through daughterless generations, a process statistically accelerated in small populations. The Y chromosome equivalent, the patrilineal Y chromosomal atom, lived approximately 200,000 to 300,000 years ago, and similarly represents not the only man alive at the time, but the only direct paternal line with continuous male descendants to the present. The existence of these specific, most recent common ancestors for mitochondrial and Y chromosome lineages is a mathematical consequence of population history that includes periods of very small, effective population size. Exactly what severe bottlenecks would produce. The out-of-Africa dispersal event approximately 60 to 70,000 years ago represents another genetic bottleneck. Less severe than the deep Pleistocene events, but clearly detectable and profoundly consequential for understanding human genetic diversity today. When a subset of the African human population migrated out of Africa and eventually colonized the rest of the world, they carried with them a subset of the genetic variation present in Africa at the time. Every non-African population today descends primarily from that founding group, and the genetic diversity of non-African populations is correspondingly lower than the diversity within Africa. The reduction in genetic diversity from Africa to Europe and Asia, sometimes called a serial founder effect, as populations moved further from the African source, and each successive founding group was a subset of the preceding one, is one of the most clearly documented demographic signals in human population genetics. The practical consequence is that sub-Saharan African populations contain more genetic diversity than all non-African populations combined, a fact that initially surprised non-geneticists but follows logically from the out-of-Africa bottleneck. The founding population that left Africa was a small subset of the full range of African human variation, and every population that colonized more distant territories was a further reduced subset. Indigenous Australians and Papua New Guineans, who descend from one of the earliest out of Africa dispersals, show patterns consistent with small founding populations that then expanded over tens of thousands of years of relative isolation. This dispersal bottleneck was not extinction threatening in the same way as the deeper Pleistocene events. The African populations from which the dispersers came were not themselves reduced to dangerous levels, and the total global human population probably increased rather than decreased during the dispersal period as new territories were colonized. But it represents another genetic narrowing that shaped the diversity and the disease susceptibility, the pharmacological response differences, and the frequency of various genetic traits of the majority of the human population today. The specific environmental pressures that pushed the human lineage toward extinction at various points in its history are worth examining in their own right, because they reveal the vulnerability of a species that would eventually become the most ecologically dominant animal on Earth. Climate was the primary driver. The Pleistocene epoch, the period spanning approximately 2.6 million to 11,700 years ago, was not a period of stable conditions but of dramatic, cyclical climate fluctuations driven by orbital variations in Earth's relationship to the Sun. Glacial periods, when massive ice sheets expanded across high latitudes and global temperatures dropped substantially, alternated with warmer interglacial periods on cycles of roughly 100,000 years for most of the Pleistocene. Within these broad cycles, shorter-term fluctuations produced rapid regional climate changes, droughts, floods, rapid temperature swings, on timescales of decades to centuries. For the early human populations with small, relatively immobile populations dependent on specific prey animals and plant resources, these climate fluctuations were potentially catastrophic. The African savanna and woodland environments that supported most of the human evolutionary history were not stable. They expanded and contracted with rainfall patterns driven by orbital and oceanic dynamics far beyond any individual's or group's comprehension or control. A severe prolonged drought could eliminate the water sources that both prey animals and human groups depended on, triggering population crashes across large regions. The Lake Malawi core, a sedimentary record from East Africa that preserves a detailed climate history, shows a period of extreme aridity in East Africa approximately 135 to 75,000 years ago that corresponds broadly with the period immediately before and including the Toba eruption. This sustained drought may have severely stressed African human populations independently of the Toba event, contributing to the demographic crisis that the genetic evidence implies for this period. The coastal refugeia hypothesis, the idea that human populations may have survived the most severe inland aridity by moving to coastlines where marine resources provided nutrition unavailable in the interior, is supported by the concentration of genetic diversity in certain coastal African populations, and by the rich shell-midden deposits at South African coastal sites from this period. The concept of refugeia, the specific geographic locations where surviving populations maintained themselves through catastrophic periods, is critical for understanding both why the bottlenecks weren't worse and how recovery occurred afterward. A refugium is not a safe haven in any comfortable sense. It is the margin of survivability, a location where the combination of resources, climate, and geography was sufficient to maintain a small population through conditions that eliminated populations elsewhere. For African human populations during severe arid periods, coastal refugia provided shellfish, fish, and marine mammals as caloric resources even when inland vegetation and prey animals collapsed. River corridors and highland areas maintained moisture from orographic rainfall, even when surrounding lowlands were experiencing drought. Caves provided thermal buffering that reduced daily temperature extremes, and their geographic stability provided the kind of predictable shelter that small stressed populations depended on. The genetic structure of surviving populations after a bottleneck reflects the geography of refugia in specific ways. Populations that survived in separate refugia, isolated from each other for the duration of the bottleneck by the barriers that prevented refugial populations from merging, accumulate independent genetic variants during the isolation period. When conditions improve, and populations expand and reconnect, the resulting admixture produces specific patterns in the descendant population's genetic structure that genomic analysis can detect. The complex population structure of modern African populations, the multiple distinct ancestry components that reflect ancient divergences and reconnections, may partially reflect the refugial structure of human populations during the severe bottleneck periods. The aftermath of near extinction events is as important as the events themselves for understanding the human story. Because recovery from severe bottlenecks is not guaranteed, and the specific character of the recovery shaped everything that followed. Population expansion after a bottleneck is initially slow. A small surviving population, even in favorable conditions, cannot grow rapidly when the surviving group is too small to fully exploit the available resources. The first generations after the worst of a bottleneck event involves a population too small and too spread across refugia to form the social networks that support cultural transmission effectively. Skills, knowledge, and practices that existed in the pre-bottleneck population may have been lost entirely or preserved in only fragmentary form in the surviving group. This knowledge loss during bottleneck events may explain what appears in the archaeological record as periods of cultural simplification or stagnation following demographic crises. Complex tool traditions that required specialized knowledge to maintain may have been reduced to simpler technologies when the specialists who maintained them died without fully transmitting the knowledge. The recomplexity of material culture visible in the African Middle Stone Age record after the post-Toba period may reflect not just technological innovation, but the recovery and elaboration of skills that had been retained in skeletal form through the bottleneck and were expanded as population sizes recovered. As populations grew past the immediate bottleneck phase, specific dynamics of expanding populations created selective environments that differed from both the stable pre-bottleneck conditions and the severe stress of the bottleneck itself. Rapidly expanding populations moving into newly available territory encountered new prey species with no evolved fear of humans, new plant resources untested for edibility and processing, and new pathogens to which no immunity existed. The selection pressures of expansion after near extinction were intense and diverse, potentially driving rapid evolutionary change in exactly the period when human cognitive and behavioral capacities show the most dramatic elaboration in the archaeological record. The philosophical dimension of the near extinction question is the one that tends to arrive late, after the numbers and the science have settled into place, and it arrives with unusual force. Every person alive today is the descendant of individuals who survived when others didn't, who happened to be in the refugium rather than the drought zone, who happened to have the immune response that handled whatever disease was circulating, who happened to find food when food was vanishingly scarce, who happened to have a child who survived when most children didn't. The genetic bottlenecks in the human past are not abstract statistical events. They are records of mass mortality among human beings. Individuals who lived, who were embedded in social groups, who presumably had something like the emotional and social complexity that modern humans have, and who died without contributing ancestry to anyone alive today. The 1,280 breeding individuals at the minimum of the deep Pleistocene bottleneck were not data points. They were people, or the closest thing to people that existed 850,000 years ago, living in a world that had contracted around them, surviving in the only places where survival was possible, carrying the genetic lottery ticket that their descendants would eventually cash in the form of the biological and cultural complexity that produced modern civilization. The line from that minimum population to the current 8 billion is unbroken, but it passed through narrows that should, statistically, have been impassable. Populations of that size don't survive 100,000 years routinely. Inbreeding depression, stochastic extinction events, disease, and environmental catastrophe combine to give small populations poor long-term survival odds, even under conditions less extreme than the Pleistocene offered. And yet, the line held, the chain didn't break. At every point when the surviving population was small enough and vulnerable enough that extinction was a realistic probability rather than a theoretical abstraction, enough individuals survived and reproduced that the lineage continued. The population geneticists who calculate these bottleneck sizes, the paleoanthropologists who find the sparse fossil record of these minimum population periods, the geologists who date the volcanic layers that mark the crises, all of them are, in some sense, tracing the outline of the narrowest passages in the story of human existence. The passages that could have ended the story, but didn't. That it didn't end is why you're here. That you're here is why anyone can know it almost did. The near extinction of ancient humans is the most important thing that almost happened in the history of the species that eventually learned to worry about such things. It is, by any reasonable measure, the closest thing to a miracle that science has ever found written into the human genome.